But today we have Dr. Luke Gregory, who is a postdoc at Cornell University, and he's going to be talking with us today about crop physiology. And that's sort of the intersection of crop physiology, AI models, remote sensing, all of this sort of truly integrated work that we're trying to achieve here with AI Leap. Please take it away, Luke. Thank you.
Yeah, thank you. Perfect. I have two monitors, so like if you see me, my eyes like drift over here, to see if anyone's like in the chat, just no, it's okay. But yeah, uh it's a very cool opportunity to talk to you guys about crop physiology. It's something that I feel like I got my PhD in plant molecular physiology. So yeah, I'm just very excited to talk to you guys about this today. So just to kind of sort of set the stage about what to expect in this lecture is, yeah, like Natalie was saying before, it's kind of setting the stage like how does this all kind of connect with the AI-leaf discipline and these sort of four curriculum pillars that we kind of stated in the very beginning, from biology to remote sensing to programming to, leadership and communication and ethics. We'll then kind of jump into sort of the main lecture components, where we'll really be trying to answer these questions of, how do plants really capture environmental resources and transform it into biomass? And then kind of taking a deeper look into these specific physiological processes that coordinate these fluxes, light, water, CO2. There's many others, but we'll just focus there today. 'Cause plants kind of just are kind of stuck, wherever they are, wherever they, emerge from the ground, they're kind of stuck there and they only have the, resources that are surrounding them in their ambient environment to kind of take a hold of and create life from. And then we're going to kind of end and bring it back to sort of, thinking more interdisciplinary about how our kind of new and our understanding of crop physiology can help us address these grand challenges that we see in agriculture. So, so we're up to today. So when we think of plant physiology, crop physiology, you know, they're all plants. But it's this branch of plant science that investigates these internal processes of plants, spanning from all scales of size to aspects of time. There are reactions that can happen in plants. I know sometimes we see them as just this green backdrop to life, but there are these reactions that are happening at the femtoseconds, and also on the other extreme end, trees can survive for centuries. So it's really interesting to take a deeper look and study. And when we think about these curriculum pillars that we've sort of established for ourselves in this group of AI-leaf undergraduate scholars, agriculture and forestry, remote sensing and integration, programming for AI, and leadership, ethics, and communication. Plant physiology, falls into agriculture. And it really provides the biological foundation that's kind of essential for interpreting what we're remote sensing, what we're modeling, and what we're managing in these agro ecosystems. So if you remember Praveen's talk, I think it was 2 weeks ago, we were looking at satellite images of corn and of soybean. And, this is going to provide some context into like, we're imaging plants, but like what's going on? So I kind of wanted to start with the question. So if you're comfortable, you can answer or let me know in the chat. But when we think about, if I went out and planted a tree sapling today and returned to it, decades later and came back to it as this sort of massive tree with all of these leaves and all of this, biomass in the trunk and root structure, when we think about like, where's all that new mass coming from? You know, do people have an answer for that? Or, in the chat, can you kind of, let me know if you think it's, all of this mass is coming from the earth or soil, this physical medium that these plants are growing in, whether it's coming from the air, whether it's coming from the light, sunlight, water, or, maybe some type of combination. Um, between these. So I'll just give that a few seconds. If anyone's brave. Yeah. Yeah. So I'm not going to tell you right now, but we'll see if we can come up with the answer. So, but we have people sort of saying that it's going to be a combination. So, so when you think about, I kind of hinted at this in the beginning, but like plants are just, they're sessile. They're wherever they're growing, wherever they emerge, that's kind of where they exist. And, when we think about these resources and where they're located around in the environment, and, really thinking about how plants are accessing them, for water, it's typically supplied through rainfall, or maybe you're in, a farming operation and it's provided by irrigation. But once that falls and hits the soil, it really forms this medium that connects both the soil, the roots, and the atmosphere together. Um, and depending on what's in the soil, if there's soluble nutrients, they can dissolve within this water. Uh, when we think about the earth or the soil, when I'm really, thinking about who's the physical medium that plants are grown in. And, I think, at least in my head, thinking about this two things. The first is that it's this dynamic reservoir for water. Water's stored up in these pore spaces and sometimes like really tightly bound to these, in between these soil particles and spaces that roots will kind of find their way to uptake water. But also they contain minerals and nutrients, so things like nitrogen, magnesium, potassium, phosphorus, calcium, a lot of other things, both soluble and insoluble. And these, are either occurring naturally, or again, if you're in a farm system, um, like you would typically be in agriculture, they could be added through fertilizers. When we think of light, it's typically supplied by the sun, or maybe you have an artificial light, in your house, or something like that. But it provides this sort of continuous stream of photons, something that, a beam of light. And a photon, it's kind of this package of a type of wavelength that can be absorbed by a plant. We'll talk about that in a little bit. But it's, the sun is this, provides this electromagnetic spectrum, including ultraviolet, visible light, which you and I can see, and also infrared wavelength. So all of that's kind of raining down on a plant. And, this is kind of where that comes from in their environment. And the last thing is air, or, this atmosphere. And in our atmosphere, it contains gases such as oxygen, which exists at 21%, nitrogen, which is around 78%, carbon dioxide as well. This is increasing, but right now I think it's around 0.042%. And there's also other trace gases that exist in our atmosphere. But when I'm thinking about what a plant's doing, I kind of separate it in, these types of physiological mechanisms that have developed into two categories. There's this sort of material acquisition where we're bringing in materials that we're going to talk about right now. And this is kind of transforming these environmental resources that, are these raw, exist kind of in these raw forms, and transforming that into a usable form of energy and matter that a plant can utilize. So when we think of atmospheric CO2, plants, through this process called net carbon fixation, have the ability to like kind of pop that carbon off of CO2 and turn that into sugars, or, more specifically like carbohydrates, or, these really elegant carbon skeletons that plants can use to build their tissues and are really the foundation of plant biomass. When we think of light from the sun, plants do a very good job of absorbing this light and turning it into usable energy. We'll talk about this in a little bit. There are these two forms, NADPH and ATP, but they're really just these two different flavors of these molecular batteries that can power a plant. Water, whether it's coming from rainfall or irrigation, can be absorbed by the roots and really acts as this sort of transport medium that moves nutrients as well as other molecules and sugars throughout the plant. It also does a lot with temperature regulation as well. And for nutrients, I've said this before, but roots can take up these soluble nutrients that maybe are dissolved in water, while these insoluble nutrients, they have to be released from soil particles before they can be absorbed by the roots. So through this gain of these resources, CO2, light energy, which or light that gets converted to chemical energy, water, nutrients, plants can redistribute all of this carbon, energy, and nutrients through various metabolic conversions that support diverse plant function and all of these physiological processes that we're going to talk about. Whether it's into growth, defense, storage. Once plants have this material, they can do a lot with it. And, whether that's production of leaves, stem, tree bark, root structures, all of that. And then there's this other component, this material loss. We're not just gaining all of these components and, expanding exponentially. There's also material loss. And plants either are releasing these or expending these captured resources through other processes. So carbohydrates, which are production of photosynthesis or this net carbon fixation, can get respired to produce energy, but a product of that is CO2 that gets put back out into the environment. Maybe you have insects or other pests that have herbivory, where you're actually losing material. You could have leaf shedding, where again, you're losing material, or root exudation, where these roots are actually sending signals in the soil, whether they're organic or inorganic, and you could be losing, these carbon structures or these nutrients through these types of material loss. And also water, it enters through the roots and moves through your entire plant, but it's transpired or evaporates in the leaf. And that water just goes right back out into the atmosphere. So when we think about defining biomass and what that is, it's really this difference in mass balance between what materials that we can acquire from our environment through all this, very fun and exciting physiology, and what materials are lost, whether that's through herbivory or, these processes that are occurring all the time, like transpiration or mitochondrial respiration that release CO2 or water. And there's other processes here, but these are some of the big ones. So biomass is just, how much do we gain subtracted by how much do we lose? So it's this sort of net gain of materials from our ambient environment. So when we think about, where did all this new mass come from? Technically the answer would be carbon from the air, because that's really the main building block for plant biomass. But to be honest with you, I would say it's probably a combination of everything, which a few of you keyed in on right away. You know, yes, carbon's being, extracted from CO2, but that couldn't be transformed into these sugars or carbohydrates without, the energy that, light provides, or, these nutrients that are a part of these carbon skeletons that are a part of all of these building blocks. So it's really this combination. Now, where we're going next is kind of more based on the title of this lecture. When we think as crop physiologists, one of the big things that we want to do for farmers or for stakeholders is to optimize these physiological processes that are involved in material acquisition or material loss, excuse me, so that we can maximize our biomass gained. So, how do we increase our agricultural yield or get, bigger corn cobs? And a lot of this, it can be complicated, but, a lot of this is just getting more resources from our environment and allocating them to these, certain, or partitioning them to these certain processes that create yield or create, corn in a specific case. So one of the things that I want to talk about, um, just to kind of get a little bit broad for a second, is really when we're trying to understand these physiological processes, we first need to understand like the unit. Um, yeah, I think I've talked about this or said this a few times, but this flux, and it's this unit that we use to describe material, moving from one space to another. So it's some amount of material that moves through a two-dimensional plane, and it has some type of time component associated with it. So if I left my window open, it could be the number of bumblebees that move through my window in a given day. It could also be an amount of photons or light that's hitting a meter squared of leaf area in a given second, or micromoles of CO2 per meter squared of leaf per second. So we don't have to get super bogged down with, what micromoles means or millimoles or milligrams. It's just an amount of material that's moving through a two-dimensional plane. And in a lot of the time, in our case, as crop physiologists, we're thinking about it as leaf area. So a meter squared of leaf area. And we can think about this as, this packet of light energy travels from the sun, in space, um, to the earth. It'll strike a leaf surface and penetrate through multiple cell layers. So here I kind of have what a typical cross section would look like of a leaf. And what I'm trying to depict here is that we have, let me see if I can get a laser pointer. But we have the top of our leaf, the bottom of our leaf. We have all of these different cell types. This is our vein. That's sort of the plumbing where water comes from. And, we have all of this, light that's kind of hitting or penetrating into our leaf at the top layer. And, some fraction of this light will penetrate into a single cell. And one of these cells they have these organelles called chloroplasts, and they are jam-packed with these other membranes called thylakoids that are what are absorbing light. So, if we zoom in a little bit further, some micromole per meter squared, um, or micromole of photon per meter squared per second will be interacting with a thylakoid. And these thylakoids, they're kind of jam-packed with all of these different photosynthetic machinery, these different proteins, but really we're not going to worry about any of that. And we're just going to focus on these light harvesting complexes, which I have highlighted in this sort of dark green. And that's really the place where these pigments are located, called chlorophyll, and their job is to absorb light. And they do this really well in the blue wavelengths and the red wavelengths. They don't really do it well in green, so that's why plants reflect back green light. But they'll absorb, light that is hitting them. And, just to kind of go a little bit further to the end, what will end up happening is once light is absorbed, there'll be water located in the interior of this, green pancake, this thylakoid membrane. And the light will basically split water. So H2O, there's two hydrogens connected to a oxygen, and there's enough power to split that and release these electrons. And these electrons will pinball through this sort of upper membrane through these different components, and it'll end up on attaching themselves to this compound called NADP plus, and with the electrons, it'll be NADPH. And we can just think of this as a sort of a rechargeable molecular battery. So in our case, light energy charges the battery, providing these electrons from water. And the plant can then use this NADPH um, for any process that it needs to, because it's sort of the storage of these electrons. Um, and a classic example is that this goes into helping build sugars from CO2. And after this energy is released or expended, it'll, fall back into becoming NADP plus, and it can come back and be recharged again by more electrons in your thylakoid. This sort of second part of this is that once water is broken, it moves into these hydrogen atoms called protons and oxygen. And this is happening all of the time in the light. And these hydrogen atoms or these protons will form a gradient within the inside of this thylakoid membrane. And at some point, they'll start moving through or powering really this protein called ATP synthetase, and it'll generate ATP or adenosine triphosphate. And this ATP, we can just think of it as like another form or another type of a battery that a plant can use. It's just kind of a different flavor than NADPH. Instead of electrons, the energy here is actually stored in the phosphate groups. And the releasing one phosphate group kind of frees a lot of useful energy that the cell or the plant can use to do work. So after ATP is released, or after a phosphate intermediate is released from ATP, it becomes adenosine diphosphate, and it can kind of come back and be recharged again back into ATP. So this is how plants go from sunlight. This light is then able to penetrate through the cell. It ends up in one of these cells, getting into a thylakoid. These light absorbing complexes absorb this light, and they use it to split water. Electrons go to create NADPH, and a hydrogen gradient forms to produce ATP. And NADPH and ATP are what plants use as cellular energy to power it. So when we think about, as crop physiologists, right, how can we optimize this process so that we can maximize the amount of light interception by a plant at like any of these levels, So I'll just kind of pause for a second and then I can tell you my thoughts in a second. But how do we think like, is there something with canopy architecture? I know here I just have one leaf, but is there something there that can maximize the amount of light we can receive? Are there different, pigments maybe that we can include in these light harvesting complexes that can absorb different amounts of or different wavelengths of light? Yeah, exactly. Leaf position. So, if we think of maize, the leaves that are at the very top of the canopy are the ones that are probably going to be the most sunlit, where the ones kind of more towards the bottom of the canopy are going to be the ones that are most likely shaded from the other leaves that are on top of it. Angle is another big thing. If you're perpendicular to your sunlight, you're going to do a better job of absorbing most of it rather than if you're at an angle. But, maybe you'd want really vertical leaves. If we think in terms of agriculture, you may be trying to put as much maize in, an acre of land as possible. And if all your leaves are horizontal, you're going to have a lot of self-shading. So it may be best to, breed for more vertical leaf angles so that you can pack as much maize or as many plants in as possible. And it's, a way that you can increase your yield by just growing more plants right next to each other because they can all share the light instead of, one or two of them taking it all and shading out the rest. So yeah, that's great.
Is that a pretty
Uh,
active area in plant breeding right now? Is that angle like
yeah. Oh, for sure. Yeah. It's I wish I had I'll find a picture and I'll make sure that we can see it next time. But it's a huge deal. Yeah. So people have identified that um if you increase your leaf angle, um, because there's probably enough light that's hitting the ground generally to provide, sorry, there's enough light that there's enough light for each of these plants. So some of them get really greedy, and it's actually competition where like you'll have more horizontal leaves so you can self-shade your neighbors so you can, compete and grow better. But in agriculture, we want, to produce, we don't want anything to compete. We want it all to, grow uniformly. So doing things like leaf angle is a big push. And if you look, and this is the image that I'm gonna find and we'll make sure it's available, is if you look at the eras, from like 1985 to like 2015 or even like 2025 today, you can see how, these hybrids that are getting produced by these companies, their angles go, more horizontal to vertical. So we're going to transition now and talk about water and its sort of journey through the plant. Um, and we're going to start just sort of with the soil profile. So at least when I'm thinking about this, I think of it as in two zones. We have our groundwater or our water table at the very bottom, which is 100% saturated with water. And then we have this zone on top of it, this unsaturated zone. And what we find is that, soil water is extremely dynamic and it's constantly being redistributed by multiple hydrological processes. So whether we have rainfall or irrigation, that water is infiltrated into the soil. Part of that could also be surface runoff. This water is redistributed. Part of it, goes into our groundwater or our water table to recharge. Some of it comes back up in capillary rise. We have soil evaporation, and it's very dynamic. And when we have roots in, these soils as well, they also do a great job of reshaping soil moisture. There's uptake of water from roots that adds to sort of this dynamic soil water system. And when we think of this root soil interface, it's really this critical zone where you'll have like a root hair or that is interacting with these soil particles. And, within kind of a soil, there's these solid structures that are the actual particles. There's water typically and air, or, parts of our atmosphere that are trapped. And it's really the root hair's job or the root's job to kind of find its way to where the water is located and absorb it. And we'll talk about that in just 1 second, because I think in this next slide or two, it'll all kind of make sense. When we kind of think about this is an entire system, we have our groundwater, we have this unsaturated zone. We can think of it as saturated. I drew this like thinking like we were up here in Ithaca, New York, where it's a glacial till. And we have these like rocks in our soil as well. But if we imagine that this top part is saturated, water will be absorbed and it'll move in an unbroken column from our roots or from the soil all the way up the stem through sort of these different branches and it finds its way to the veins of the leaf. And from there, it'll go from liquid water to water vapor and evaporate from our leaf. And there are these pores called stomata, and that basically facilitates water moving from internal to external to the atmosphere. And when we think about this water evaporation, um, in these sort of spaces within our leaf, they really create like a suction, kind of like tension. And it's really this tension that pulls the water up in sort of this column from the roots through the xylem, which is this material that's in the stem that facilitates water movement, and into the leaf. And the best way that I've ever heard this described is it's kind of like when you're drinking liquid using a straw. Like you can put your straw into, a glass of water or, pop or soda, but really to get that water to move through the tube, you have to like create a vacuum. And, in a straw's case, it's happening, through your lungs. But in a plant's case, it's happening through this sort of evapotranspiration that's happening at the leaf level. And that's kind of what's pulling this water up. And water is good because it also has this adhesion and cohesion properties to it. So it's very good at kind of, you know, moving through. Now, again, we're going to zoom in. We have this vein, which I kind of depict here with all of these different cells around. This is the top of our leaf, this is the bottom. And water will evaporate and go from a liquid to a gas. And it'll just go through passive diffusion. And eventually it'll find its way out into the atmosphere. And this is driven mostly by a concentration gradient. So, and it's really driven by dry air that'll pull kind of the water vapor out. And you'll hear this term called vapor pressure deficit. It's really, when we think about it, our internal leaf, we can think of it as like a wet sponge that's, 100% saturated. But, outside, at least, here in, New York, it's definitely not 100% humidity. And that's kind of the idea of what vapor pressure deficit is. It's the difference in vapor pressure between, the internal leaf versus, what's in sort of the ambient atmosphere. And we can think of this, at least the way that I was taught, is kind of like Ohm's law. So there's, a current, there's a voltage difference, and there's your resistance. And when we kind of think about this more in biological terms, transpiration is our current that's moving water instead of electricity. Our vapor pressure deficit acts as this voltage difference. And this resistance to water, which is facilitated by our stomata, is acting as this resistance. But in a lot of the times in our case, we don't really call it resistance. It's the inverse. So it's 1 divided by resistance, which is conductance. But you're either saying, how easy is it for water to diffuse through, a this pore, which would be conductance, or how much resistance does it have? But most of the time we're using conductance. So yeah, so when we kind of think about, when there's all of these periods, when we have water or, these plants are well watered, we never really have to worry about water as being a limiting factor. But there are times that, plants are either in drought or we have these heat waves that, are anomalies, but it doesn't really seem like they're anomalies anymore. And it's really this stomatic conductance, this sort of pore that really facilitates if a plant can preserve water or if it, is just hemorrhaging out water, if it's completely opened. So, what you'll end up finding is, in desert conditions or, if temperatures are really hot or in drought conditions, plants will really try and close or lower their stomata conductance to kind of preserve water within the leaf until they have enough water to open it back up. So again, I'll ask this question: is crop physiologist, how do we think we can optimize water use so that plants can have enough of it to support growth and and stay hydrated, I suppose, but while also kind of minimizing unnecessary loss? We kind of talked about the stomata and how it can fluctuate uh, to either preserve or release water. But are there any other things, whether it's root related, or anything else that we think, that we could think about how plants could, optimize this process? Whoa, sorry about that. Yeah, so humidity. So when it's really humid out, um, uh, your when it's really humid outside, or that's kind of the environment that your plant is existing in, it's losing water isn't that much of an issue because there isn't a very strong deficit that's going to be pulling water out. But for plants that don't live in humid conditions, it can be really challenging. And I'm just going to kind of move into this next step because we'll see how hard of a job stomata have it. So part of the issue, so we're now going to kind of talk about the journey of CO2. So carbon dioxide is in the atmosphere all over, and I think it's around 0.042% of our atmosphere. And the same way that water diffuses out through the stomatal pore, CO2 can diffuse inward as well. And it can kind of move through, same passive diffusion, until it sort of finds itself um, diffusing into a single cell, into the chloroplast. It doesn't go into these sort of thylakoid membranes, but this reaction happens just outside of them in the chloroplast. And you end up, the CO2 diffuses to this enzyme called rubisco, and it's really this initiator of carbon fixation. And when you have CO2 and this other substrate called RUBP, your enzyme rubisco can facilitate this carboxylation that basically pops off carbon from the CO2 and attaches it to RUBP. And that is kind of the first reaction in many that facilitate this downstream carbon metabolism. And that's what creates your sugars or these sort of carbohydrates, starches and sucroses that really go to fuel, the build-in of these different compounds that, are making leaves or making stems, tree bark, roots, all of that. So when we kind of go back and think about the role of the stomata, when when the conductance is high, all of this CO2 can come in. And maybe in this case, like you were saying, humidity here, if this is in a humid environment, this would be wonderful because you wouldn't really lose that much water to the environment, but you could maximize your stomata conductance where you could, have all of this diffusion of CO2 come in, which would increase your CO2 molecules that end up around rubisco, and you could do, and this would just facilitate this reaction to happen without any type of limitation. But if we're in an environment where maybe there is really low humidity and the plant needs to conserve water, and what will happen is the stomata conductance will get really low, and the consequence of that is that because you're lowering the stomatic conductance, there'll be less CO2 that can diffuse into the interior of the leaf and get to rubisco. So you're really going to limit how much carbon can be fixed. And this will have a growth and yield impact. So, it's a hard thing, right? Stomata face this major dilemma. It's either death by thirst, where you have your stomata pore completely open and you're just hemorrhaging out water, but you're maximizing, CO2's ability to get into your leaf, or you're dying by starvation, where you're closing, essentially, or lowering your stomatic conductance to preserve all this water, but you're really not allowing that much CO2 to come in. So my question is, how do you think that plants, you know, optimize this process? Because it seems really tricky that, the CO2 partial pressure, the amount of these molecules that can end up around Rubisco, we want a lot of them so that we can, do our net carbon fixation without kind of any limitation, but we don't really want to, we're trying to minimize unnecessary water loss. So, does anyone have any ideas of like how maybe plants could facilitate that?
Better regulation around stomatal opening. Is that
Yeah, so it's a very tough job. Like I wouldn't want to be a stomata, right? Like they're constantly adjusting, as the environments, even in a day, they're going from, open to shut to, always changing to account for, temperature or humidity or vapor pressure deficit, sorry. So it's really tough, and people are really. We'll get into this in a few slides, but people are really looking at this stomata for the answer. But the way that plants do it is because they really can't change, the physics of diffusion. They end up changing their biochemistry. So we don't have to get super concerned about this, but in terms of like these categories, but C3 photosynthesis is kind of our default, which we described, where CO2 just passively diffuses into our leaf and diffuses to rubisco. But there are these other two pathways that have developed actually in high temperature environments where your plant's always going to want to stay hydrated. So it'll always, well, there's always exceptions, but for the most part, it'll always sort of keep a lower stomatic conductance. And when CO2 diffuses in, they build these what they call carbon concentrating mechanisms to kind of grab CO2 and shift it around into different cells to concentrate CO2 around rubisco. And the reason that this matters is when we're thinking about putting together these crop forecasting crop yield and these different models, whether we're dealing with corn, which is kind of this center one, C4 photosynthesis, or like soybean or wheat, which kind of have the C3 photosynthesis, these this biochemistry changes. So when we're like making these sort of mathematical descriptions or equations to kind of describe this biological phenomena, they're going to be different depending on which type of photosynthetic metabolism that a plant is going to use. So I know that we have maybe less than 15 minutes left. So we're kind of at the end where I just want to take a second to talk about the grand challenges and how, this crop physiology knowledge can kind of help us solve. So we can use, crop physiology, to make these mathematical models to describe growth phenomena in plant systems. We can, understand crop resource use efficiency, whether it's water, light. We didn't really talk about nutrients or anything like that, but we could start to think about where the limitations are. We could optimize farm management practices. Maybe we could even predict crop growth dynamics and yield. Um, and then another big thing is breeding for sort of these climate resilient and high yield crops. And that's kind of where I want to talk about for the rest of the lecture. Um, for plant breeding, just like this sort of theoretical understanding of crop physiology that we kind of worked through earlier isn't really enough. The challenge is actually like capturing these traits of interest. So how do you capture plant height or leaf area or these things that we really think are important, leaf angle, right? Um, to capture them, to compare them across, breeding populations so that we can pick the best one. And that's the challenge where we need to leverage robotics, sensors, programming. So when I think like what, like you all, like you'll have your domain expertise from, your university, but you'll also have kind of these, curriculum pillars where you'll have this interdisciplinary perspective and this broader perspective of these challenges. So if I was so maybe there's a plant breeding team and they say to you, can we develop a plant variety, maybe it's maize or soybean, that has reduced water loss, and we think we can do this by modifying either stomata density, the size of the stomata, or like something about its geometry. Typically, the ways that you would go out and measure this is, and I'm sure there's maybe a little bit more clever ways nowadays, but you have like clear nail polish that you can paint on the back of the leaf. After it dries, you can peel it off and put it under a microscope, just like a light microscope that you can buy off the shelf. And you can go and count, these stomata. And maybe for a certain, known area, you'll count the density of them. And this is very subjective. It's time consuming. It's really labor intensive. And like, we do not need to be bogged down by this. So, you know, very recently, like this is a paper in 2024, but people, are applying these techniques that we're talking about to really help streamline these analyses so we can look at way more, samples um, than we could before doing it manually. Capturing the images, processing them, doing this sort of network training, and then looking at, detection or segmentation and these types of things. So again, I have these papers here. The goal really isn't to really talk about these pipelines, but just to show you like people pretty recently have been doing this where they're developing this, whether it's YOLO or CNNs or this programming in this architecture to detect, segment, and measure, these anatomical features that we can get in these stomatal imprints. Another thing that you might find, we didn't talk about disease pressure, but you may have plants or plots that present with some type of disease. And what people end up doing currently is they'll train scouts to go out and manually scout plants. And again, it'll be on a scale of 1 to 5. How severe is it? Presence, absence. And, maybe there's a better way to do this. And what people have found is that first you have to identify which, disease is actually northern leaf blight. I certainly don't know because I'm not that type of biologist, but it's this one on our left that has these sort of cigar-shaped lesions. And people have developed these convolutional neural networks as this alternative to manual scoring to detect presence absence just from RGB images, whether these lesions are there or whether they're not there. And if you want to read more about this, you can go to this paper. And maybe you say, maybe I don't have to go through this cornfield with my smartphone. Maybe I can just attach, an RGB camera to a drone and fly it at a low altitude to collect these images that I can, read into this type of um, algorithm to let me know if there is northern leaf blight or not. And the other one is, maybe you have plant breeders who are interested in developing short stature corn. This is a huge accomplishment. It's been done in companies in the Midwest where they have these really severe windstorms that kind of topple these corn plants because they're top heavy, and they really wanted to develop short stature corn. But maybe, the way that they're doing it or the way that they're measuring plant height is by having these barcode measuring tape and these scanners that they currently go out and, scan to get plant height. So this is more of an open question, but like what tools, technology, sensors do you really think you can leverage to measure plant height in a high throughput and reproducible way? Could you take advantage of drones? Would you think, maybe you could do something with LiDAR, or maybe you could use multispectral imaging or just RGB imaging? But how can you make this process more efficient? So that's kind of where I see, crop physiology intersecting with application, especially in a breeding kind of sense, where you're just trying to find these traits that you can extract from either images or LiDAR or whatever sensor data that you're dealing with. And yeah, I know for me it felt like it was long, but in terms of like the summary of what we covered, we kind of spent the first part talking about how plants capture environmental resources and transform it into biomass. We learned about, atmospheric CO2, photons, water, nutrients, where they're at, and how plants access them. We spent the bulk of the lecture talking about really in depth about these physiological processes that coordinate photon fluxes, water fluxes, and CO2 fluxes, and how really important the leaf is at doing this. And then we had just a few case studies that we kind of flashed through at the end, where we kind of, took what we learned and tried to make sense of it in some of these case studies in a plant breeding context. So thank you. Let me know if you have any questions.